Well, thank you so much for inviting me here. It's really a pleasure and honor to be able to share some of our work. The goal of this talk for me is really sort of multifold. I want to introduce our amazing Kyle DeWardies, who are going to be talking about their research. I want to talk a little bit about my own work in Waldenstrom's macroglobulinemia. And I want to also provide a bit of a historical context so people can understand how this work fits into the larger picture of Waldenstrom's macroglobulinemia and sort of where things are headed in the future. And lastly, my goal is to really emphasize the outsized role that the IWMF has played in funding this research and making all of this progress possible. So my name is Zachary Hunter, and I'm the Associate Director at the Bing Center for Waldenstrom's Macroglobulinemia at the Dana-Farber Cancer Institute. And I do all the genomic and bioinformatic work in this for our group here. So for a bit of historical context, I first joined Steve's lab at the time in 2003. I had been out of college for a little while and was thinking about applying to grad school, and he convinced me to work for him for 1 year. And obviously things have gone on since then. But at the time I joined, it was just me, Andrew Brannigan, who's now gone on to be a very well-known physician at Mass General Hospital in Waldenstrom's and multiple myeloma, and a volunteer named Bob Manning, who's an amazing guy. And that was the entire center. And at the year I joined, there had been several papers actually that came out looking at overall survival in Waldenstrom's, both coming out of Europe, both placing the overall survival around 6 years, just a little bit over. And the therapy back then was very different than anything we see today. It's very early chemotherapies, and the monoclonal antibody rituximab had just been introduced and coming into widestream usage. And which is, again, just to give you a context of how much things have changed since where we are now. There was a big debate going on at the time about whether Waldenstrom's was even a thing. There was a lot of debate about whether Waldenstrom's was actually multiple myeloma or some other B-cell malignancy, and it was just a weird variant, and that we shouldn't be paying as much attention to it. But the one bright spot is that Stephen Trion had just the year before hosted the first ever large gathering of Waldenstrom's clinicians and scientists to start to come to a consensus about what WM was, how do we diagnose it, and how do we start talking about responses to therapy. So everyone could get on the same page and we could actually start making some real progress in the disease. So state-of-the-art research back then was pretty basic. My first project was actually working on characterizing familial history in Waldenstrom's macroglobulinemia and looking at correlations with these large-scale deletions in chromosome 6q. Now, I didn't find these deletions. These were first described by Fonseca and others at the Mayo Clinic. But this was kind of all we knew about Waldenstrom's at the time, which is not very much. Now, I have gone on to continue to work on familial history in Waldenstrom's, and I'm going to be talking about that in the Sunday session for those of you who are interested in learning more about that. Now, once I was clear that I wanted to stay and continue doing this kind of work. Sure. Is this better? Awesome. All right. So once I had decided to stay and really dive into this kind of work, I figured I should take it seriously. So I actually applied to a PhD program at Boston University, and my dissertation was on whole genome sequencing in Waldenstrom's macroglobulinemia. And this ultimately led to the identification of the MYD88 and CXCR4 mutations that we now know about in Waldenstrom's. And thanks to the work of really people from all over the globe, we now know that these mutations, like for instance, the MYD88 are in about 95% of Waldenstrom's patients, and CXCR4 in about 40%. So slightly before this happened, Steve had noticed this drug that was in development that was inhibiting a gene called BTK. And he was convinced that it was going to be a really important breakthrough for Waldenstrom's. Now, we didn't know anything about these mutations yet, so he went and he asked, look, can we use gene expression to try to make a prediction about this? And sure enough, we could see that this pathway was sort of overexpressed in Waldenstrom's. And based on that data, we were able to get a clinical trial going. Now, once that trial was going, we made these discoveries about the mutations, and people started digging into what exactly these mutations meant. And in both cases, we found that these mutations were driving constitutive activation of their respective pathways. In the case of MYD88 is part of a normal biological process where cells detect bacteria and viruses. And they pass that signal on down and start an inflammation process we call NF-kappa B. And this NF-kappa B is really important for Waldenstrom's growth and survival. And what Guang Yang from our group was able to show is when we block MYD88, that NF-kappa B goes away in Waldenstrom's. And more importantly, while MYD88 was thought to signal through the Iraqs, and it does, the actual NF-kappa B signaling in Waldenstrom's was being driven through this novel association with Bruton's tyrosine kinase, or BTK, which is the target of ibrutinib. Well, this, of course, made us stop everything, and we went and looked at this clinical trial, and sure enough, those people who had mutations in MYD88 were responding beautifully. People who lacked the mutation weren't signaling through BTK and were not responding to the therapy. So we could actually make a prediction about who would respond and who wouldn't. Not only that, we looked at the CXCR4 mutation status, and we discovered that people with CXCR4 mutations were responding very well, but we were not getting the same depth of response, and the speed of the responses were much slower. And I think some of you have heard a little bit more about that in the previous session. That was really nicely done just in the session previously. So with this information, we were able to go to the FDA and say, hey, look, we have these amazing clinical responses. We have this amazing genomic data. We can make predictions about who's going to respond. It all kind of makes sense. And based on this, it's Waldenstrom's that actually got the first ever breakthrough designation, not colon cancer, not lung cancer, not multiple myeloma. Waldenstrom's got the first ever breakthrough designation approval for the FDA. And they announced it on Sutrion's birthday. So we haven't stopped with MYD88 and CXCR4. There's been a lot of interest in trying to understand how BTK inhibitors work, how they work differently in different patients, and what causes them to ultimately fail. And this is actually some of the work that's going to be talked about by Dr. Hao Sun from our group, using single-cell sequencing to actually look not just at the WM cells, but the bone marrow overall, and look at these changes to make really interesting predictive measurements about how these inhibitors are working in WM. So as our biological understanding started to really improve a lot with the discovery of these mutations, there was really an explosion of research and also of therapeutic advances. So not only do we see BTK inhibitors and new BTK inhibitors, now over the course of this conference, I think you'll hear about new degraders coming out targeting this pathway, upstream inhibitors. We also have proteasome inhibitors. We have BCL2 inhibitors targeting cell survival. We have IRAK inhibitors, which are in development, targeting the other side of this MYD88 signaling cascade. And we even have done clinical trials on CXCR4 inhibitors showing that they greatly benefit people who have CXCR4 mutations. So this really changes the therapeutic landscape for Waldenstrom's. Now, it's not just limited to the clinical side. Basic science, too, is really starting to explode now that we actually have a better sense of this underlying biology. And I just wanted to highlight two of last year's speakers at this session, Philip Garberitz and Tina Bagratuni, who I just saw very recently, one in Athens and one just back at the Farber within the last few weeks. And both of them independently told me what a big deal it was to get this award and the money that came with it, and how it helped them really shift their focus from working in both cases actually on multiple myeloma and shifting to be able to work in Waldenstrom's. And the reason why this matters is that the two of them have really become rock star scientists who are doing amazing work. And again, this has been made possible through the support of the IWMF. Now, as for my own work, I've been working a lot on trying to understand the molecular characterization of Waldenstrom's, what's going on and why and how does it evolve. And this led to the discovery of two distinct subtypes of Waldenstrom's that we call B cell-like and plasma cell-like Waldenstrom's. And we've been able to show that this sort of evolves from a signature that we call early WM that corresponds to MGUS and smoldering WM. And these two different subtypes, they have very distinct mutational profiles and signaling profiles. And we're using this to try to understand more about what makes Waldenstrom's tick and what we can do about it. Now, I'm not the only person who's doing this work. There's also Patrizia Mondello, who's done some amazing work looking at, sort of coming at it from a different angle, looking at different WM subtypes. And we actually now have a grant together to integrate our two findings and try to better understand Waldenstrom's macroglobulinemia. She's actually here at the conference. If you see her, she's amazing. You should definitely stop and say hi. Now, the other thing that we've been doing in addition to subtyping is looking at something that we call the WM evolutionary score. And this is a molecular score that allows us to look at the degree of progress that WM samples have made from smoldering Wallenstroms to progressive disease. And the reason why this is helpful is once we were able to dial this in, we could start imputing it into new data sets that weren't ours. So this is actually a data set from Italy, looking at IgM, MGUS, and Wallenstroms, and you can see the two subtypes clearly in red on each side, and the MGUS at the bottom. And once you can start to stratify these groups, it's easy to come up with molecular signatures. And these signatures give us insight to WM and allow for things like improved diagnostics. And this is, again, Philip Garberitz in a collaboration that we're doing, showing that we can use things like immunohistochemistry to tell them apart. But also, the genes that are involved are really critical. In this case, Wink2 is a very interesting gene, and it's a gene that's been picked up and really run with by Maria Luisa Guerrera from our group. And she's really made it her own and has done amazing work showing how Wink2 drives WM evolution. And she'll be talking about this very soon. And her paper, we just found out, was recently accepted for publication. So congratulations, Maria. So the reason why we're interested in things like this WM evolutionary score is not so we can make measurements and make pretty pictures. It's so we can get insights into what's driving disease progression. Because if you can understand why something is happening, we can try to do something about it. And one of the real surprises we found was that inflammation was very much involved. Now, that was expected, but what was unexpected is the fact that inflammation came early and faded away. And this is very confusing to us for a while, but it actually fit into some of the work that was being done by others, including, again, Patrizia Mondello, who was looking at suppressor cells that were there in the bone marrow that were sort of silencing the immune system. These T cells that are supposed to be going around looking for cancer cells were being quieted down. And it looks like the Waldenstrom cells are actually, in these early stages, are orchestrating this development, allowing themselves to then have the room to grow and survive. And also Maria Luisa Guerrera is, in addition to working on Wink2, is working on inflammation in Wallenstroms and some inflammatory markers and how that interacts with BTK inhibitor therapies and other therapies. And both of them are going to be talking on Sunday, actually, about their respective research. I definitely recommend you come check that out. And a little fun side note, both of them are actually from the same town in Sicily. I want to know what's in the water there. It's amazing. Now, I can't talk about cytokines and cellular communication without at least mentioning Steve Ansel. He's on the board for the IWMF and has done really amazing work with the institution, but has also done a lot with cytokines. This is how cells communicate with each other. And one of the things he's always talked about is when cells see something, like a bacteria or virus, they're not just going to take that signal and use it internally to respond. They want to shout. They want to get that information out there so not only they can respond, but all the cells nearby know what's going on. And this means that the blood is a real big source of information. And this is actually the angle of research that Pieter Langerhorst is going to be talking about here in just a minute. And you can use special kinds of techniques called mass spectrometry to sort of listen in to these cellular conversations that are happening and use that to understand what is driving progression in Waldenstrom's macroglobulinemia. I'm also very excited. Pieter is actually going to be coming over to the Bing Center for a number of weeks. We're going to try to integrate all of our data sets to see if we can harmonize them and really make sense of what's going on. So very exciting research. So where are we now? WM overall survival has tripled, but honestly, it's wrong. It's completely wrong. And any number you hear is wrong. And that's because every year there are new therapies There are new clinical guidelines that are pushing that needle. From 2003 to 2006, the scenario for Waldenström is completely different. We now really have a much better understanding of the biology. And one of the amazing things is, as we start to figure things out, we can go to drug companies who have targets going after these things and be like, hey, look, this would be an amazing therapy in WM. You should come try it out, right? Before, when we didn't know what was going on, very hard to make that argument. Now we can. We also have the WMNet, run by Jorge Castillo, and also the European consortium as well, that is driving all sorts of new clinical trials to advance therapeutics. And all this is made possible by funding provided by the IWMF. And I really can't stress that enough. All this research is really made possible through roadmap grants, through the Kyle Awards and other programs that wouldn't have been possible otherwise. And in fact, we've made so much progress. We aren't just focusing on keeping people alive. We're also now starting to be able to focus on quality of life. What are the treatments that will actually not just help people live longer, but lead better lives? And this is a really big advance. Now, I can't talk about the history of Waldenström's without mentioning these two individuals. One is Robert A. Kyle, for whom the Kyle Award is named. And he's still alive. He lives and works at the Mayo Clinic in Rochester. He actually discovered MGUS and characterized it and just made foundational contributions to myeloma and Waldenström's. And Steve Treon, I know a lot of you know him and have seen a lot of his breakthroughs and advances, but a lot of people don't know is the work that he did to organize these workshops, bring clinicians and scientists together, get everyone working together and working together cooperatively. And without these two individuals, I don't think we would have been able to make the progress we have. So between the IWMF, Robert Kyle, and Steve Trent, I'd really like to give a round of applause and hope that if we clap loud enough, Robert Kyle can hear us up in Minnesota. So thank you very much. I'd like to thank the IWMF and the Tim and Ginny Bliss Investigatorship for Waldenström's, along with the Bing Fund, everyone at the Bing Center, and of course, all of you who helped make this research possible. So I'd like to start off by inviting Pieter Langerhorst to talk about his work in serum proteomics in Waldenström's macroglobulinemia. And we'll have questions and answers at the end, once all the Kyle awardees have had a chance to speak. Pieter.
Yeah, so I'll be joining you online for me, this is an evening. Thank you for hosting me online and giving me the opportunity to speak. I think it's such an honor to share my research with all of you. Yeah, so as Zach already said, I'm basically looking in blood for new disease clues regarding WM. So WM is not the same for every patient. So as some of you might know, it's a very different disease from patient to patient, and therefore it remains very difficult to predict which and when patients progress from an MGUS, which is a benign pre-stage, to a symptomatic Waldenström state, which also requires treatment. Second, it's also very difficult to predict which treatment and what treatment and what kind of strategies works best for each patient. So as Zach said, we already know quite a lot about WM. So we know a lot about the DNA makeup of the tumors, so the mutations, so the MYD88, the CXCR4, and we also know quite well in recent years how tumor cells interact with different kind of immune cells in the bone marrow. However, this cannot fully explain the differences between patients, so we need more knowledge on the processes that underlie disease progression from an MGUS to a WM and treatment responses, so which treatment works best for these patients. So, I like to study proteins in blood. I think they're so interesting and they can tell us so many things. So our cells in our body and also the tumor cells release many proteins into the blood. I think the prime example in our case is the IgM protein, which we use in diagnostics. Because so many cells share their signaling into the blood. The proteins in the blood hold many clues about actually what's going on a body-wide scale. So studying these proteins can help discover new disease clues or markers that we can use to stratify patients or better identify patients that are at risk for progression. So today I want to give a glimpse about the research that we've been doing. And we had two aims. So first, we want to see what proteins and what biological processes that belong to these proteins are linked to disease progression from an MGUS to WM. And secondly, we wanted to see how do these processes change when patients are treated and with different responses to treatment. So how do we actually do this? Well, we use proteomics. So we take a little bit of blood, so 10 microliters, that is less than a single drop of blood. Then we do proteomics. And to make it a little bit less abstract, this is the machine that I'm working with. It's called a mass spectrometer. So we do our analysis on this machine, and in less than a single drop of blood, we can detect over 800 different proteins. So these are huge data of all these proteins that are coming from neutrophils or platelets or blood cells, but also coagulation. So what makes your blood clot and in what situation, but also very important information about antibodies. So we believe that by studying this, we can get more insight. So research you never do alone. So I think it's so critically important to mention these two people. Is Wouter Verhaar, who is a PhD candidate, and Josephine Vos, who is a hematologist, both located in Amsterdam. And together we collaborated on studying our aims. So they provided the samples from their biobank, which is so important. They also provide the clinical background of the patients and helped to put everything into perspective. So we performed our analysis on these patients. And then the problem, but also the fun starts. So you have 800 proteins measured over a lot of patients, and then you have to make sense of these proteins because they're linked to biological processes. So we compared the levels of all these 800 different proteins between the disease stages, so MGUS, asymptomatic and symptomatic, and then we grouped them based on the same biological processes where they are coming from. So here's a very schematic overview of what we found. So on the bottom, I don't know whether you can see my pointer, we have controls, MGUS, so the benign, asymptomatic, symptomatic. And these represent the 4 major groups of biological processes that we found. So what we see antibodies going down, so they are decreasing, which makes sense because the tumor is suppressing the growth of your normal antibody system. Surprisingly, we also saw fat metabolism signals going down, which is kind of new, but also kind of confirming previous literature. What we saw going up was, of course, the immune system / the tumor signals, and also the inflammation going up. And inflammation, I think in all research, is so important and not well understood. So we have these signatures. We see inflammation going up, the tumor going up, antibodies and fat metabolism going down. Next, we wanted to see how these signals change when patients are treated. So what did we do? So we map the protein levels and map them to patients that were treated. And we group them based on outcome after treatment. So a minor response or a major response or a complete response. And actually we saw some surprising things that we don't fully understand. So we saw, luckily, the tumor signal going down with a better response to treatment. So indeed, it kind of gives us insurance that our technique is picking up the right signal. Fat metabolism was slowly recovering to normal-like levels. So these are the controls in our situation. And antibodies were also slowly going back to control levels, so to healthy-like levels. For inflammation, we saw a completely different pattern. So it actually seemed to be going up the better patients respond to treatment. And this is, of course, counterintuitive to what we previously saw. So we're still really puzzled by it, and we're still doing a lot of measurements, and we're still breaking our minds over what this means and how this can occur, and what does it actually mean in a patient. So what is happening? So this was a glimpse in what we have done so far. So proteins in blood are crucial to learning more about WM. And I think especially inflammation is not well understood, and we should know more about this. So we call this kind of proteomics technique hypothesis generating techniques. So we don't have an idea that we want to test, but we come up with new questions, and that is definitely the case here. I think we started with 2, and we end up with 200 different types of questions that I think will take years and years. I don't think in my life I will be able to answer all these questions, but it does highlight the need for more research into blood proteins in relation to WM cells. And the final point, I cannot stress enough that the collaboration between laboratory scientists, as me, and clinicians is key for the success of research. So what is next? And I think there is where the IWMF CAL work definitely came in. So we have so many questions, but we feel that it's so important to understand more. So in this CAL award, in the next, I think, almost 2 years, I will be diving way deeper into the proteins in blood and look specifically at the tumor genetic landscape together with Zach, as you already told, to see CXC4 mutations versus MYD88 mutations. What does it mean? Is there more inflammation, less inflammation? What does the fat metabolism do? We will study how these proteins change during treatment, and can we maybe make a prediction based on these proteins, which patient will respond best to what treatment. I do this together with Marie-José Kersten and Josephine Vos in Amsterdam. Finally, I will dive way deeper into disease progression. So together with Simundo from Iceland in the IWMF study, I will study the bone marrow proteins together with blood proteins in patients that progress or do not progress. And then we hope to find early signs of progressions, or even maybe a hallmark signature of early progression. So I think IWMF has been very important in progression of my research line and of this research line. So with that, I want to thank everyone. I cannot understate how important the involvement of patients is in this study, the sharing of their material in biobanks, their willingness to help our research further. So the study I just showed was actually funded by the Hematon, which is the Dutch Blood Cancer Patient Society. So I wanted to thank them, IWF, and of course all my colleagues at Sanquin and Amsterdam UMC, and you for your time.
Do we maybe want to have a few questions for Pieter now, because so it is kind of late over there, and then we can let him go. Is that something we could do?
I'm coming for you.
3 hopefully short but related questions. How do you measure inflammation? What kind of a scale do you use? And can AI help with this process?
Oh, okay.
I think these are very interesting questions. Yeah, so inflammation. So here it's mainly CRP, which is a known inflammatory marker. CRP in combination with SA1 and SA2. These are kind of hallmarks of inflammation. The scale, so the problem or the challenge with this technique is that it's relative quantification, so I don't get a grams per liter that a doctor can interpret. So we're trying to work out how we can make it an interpretable number. And AI, whether AI can help, yeah, I have the feeling that AI can always help if it's used correctly. I think definitely, especially if you're talking about integrating, what Zachary told is integrating a lot of different data sets and try to make sense of that. The data is becoming so large that we just cannot interpret it anymore. I don't trust myself by interpreting this data. So I think definitely in pattern recognition, so recognizing specific patterns that belong to specific patients or subgroups or treatment responses is key. And I think the research is really carried by these developments in these fields.
Am I on? Yes. I have a follow-up question that involves our frenemy AI. I wondered when you're talking about generative research and that you are creating a lifetime of questions and haven't yet arrived at a hypothesis to test. I thought that was fascinating and wondered immediately, can AI get you closer to creating that hypothesis? So it relates to the earlier question.
I think definitely yes. I think it helps us focus. It can, if we talk about pattern recognition, it can identify the features, so proteins or signatures or whatever that are important for division between patients that might progress or not progress. And therefore we can focus our research on these features instead of the millions of other features. So I think definitely it can help us in guiding and focusing our research to the most relevant things, yes.
Thank you so much. That was fascinating. And you ended on a very hopeful note, talking about the potential promise for the hallmark signature of early progression. Can you look into your crystal ball and tell us a little bit more about hallmark signatures and what might be coming down the pipes in the future?
I wish. No, I wish I had this crystal ball and I could tell. No, so at least my line of research really focuses on proteins, which are understudied in WM. So we tend to look at genes and gene signatures. So I don't know, but we're really looking into the entire spectrum. So we're looking into very early bone marrow cells that are not even decided to become a B cell or a tumor cell, to see whether we can already find something there. And then we trace it across the entire path to a tumor cell. So we hope by this sort of lineage separation, we can get some answers, or at least confirm what has already been published.
Thank you, Pieter. And it's really great to see you again. And I think next we're going to have Luisa Guerrera talk about her work in Wing 2.
Good afternoon, everybody. I'm really happy and I'm grateful to the organizer of the event to invite me to present our work. And I'm really happy also to share my joy, my excitement for the acceptance of our work that is actually funded by the Kyle Award. So my gratitude and our gratitude of our whole group goes to IWMF, because with your findings, we can actually advance the disease. Okay, so let's get into the premises of my Kyle Award. So this was a study that we in our lab in Dana-Farber conducted in collaboration with Dr. Carrasco's lab at Dana-Farber, when we created a mouse model where we introduced the human mutation of MYD88 in mice, and we observed how this mutation developed over time, so what effect this mutation actually had. So over time, while we were able to resemble some of the features of the disease, none of the mice got Waldenstrom's. So we were left with the questions, what are the additional events that actually come into play and that push the disease forward, so allow the disease to come to its full-blown context. And this is what we call second hit. Another seminal paper for landing the basics of my research for the Kyle Award was another work we did initially, where we identified Wing2 as one of the most dysregulated genes in mediated mutations. So the fundamental question that I asked in my Kyle Award was, can Wing2 actually serve as one of the second hit? So one of these events that comes in after mediated and helps the tumor progress. So now allow me to rehash a little bit back some of the amazing work that Dr. Hunter has recently made that come to a new classification of WM. So by studying RNA samples from about 300 untreated WM, he was able to introduce two new concepts. The first one is the existence of three subtypes of WM. So the first one is called early WM, and those are pretty much early phases of the disease. And then as the disease progresses, the early WM evolves into either the B-cell-like or BCL WM or plasma cell-like or PCL WM, and both of them are later phases of the disease. And then the second concept was this concept of an evolutionary score, also called E-score, that is basically a set of genes that tracks with disease progression. And this is going to be important for the next slide. So now we go into my own research. So again, basic question, can Wing2 serve as a second hit in WM? So the first way we started addressing this question was looking at our big data set of WM samples at the RNA level, and we looked at the expression of the gene. This means how much of this gene is actually available to the cells. So the way we read this graph, if you look at the y-axis, so the axis on the left, at the bottom, it means that there's no expression of the gene. And then the more you move upwards, the more the expression of the gene actually increases. So as you can see, in normal B cells that are here indicated as HDPB and HDBM, there's no expression of Wing2. But then as we moved towards mediated mutated WM, then you see that those are all highlighted. Then you can see that nearly all patients in early stages of the disease, so the early WM subtype that we just talked about, we could actually identify in this subtype of WM intermediate to really high levels of the expression of Wing2. And then as the disease progresses into later phases, the upregulation either gets further increased in the PCL subtype or it gets turned off again in the BCL subtype. So that was interesting. And then we wanted to see whether this information at the gene level was actually used to make Wing2 protein. So the way we read these figures is basically the more brown color you see, the higher is the expression of Wing2 protein. So as you can see, the highest expressors are actually WM patients. At the bottom, you see two patients that are in the PCL subtype, and one has, on the left, an intermediate expression, the other one has a really high expression. And then in the middle row to the left, you see the patients with the BCL subtype that has no expression. And that's pretty comparable to the healthy donors, which is the really top line. And you see the only cells that do express Wing2 are a specific type of cells that are plasma cells, but they are different by the B cells. So that was interesting. So right now, we got to understand that Wing2 is not expressed in normal B cells, while it's really highly expressed in WM tumor cells. This prompted us to look into the literature and understand what was known about Wing2. And what we realized is that the only study that were published were in certain solid tumors where the expression of these genes was actually turned off. And this was because Wing2 had anti-tumor functions. So the next question came pretty naturally to us. Can Wing2 have pro-tumor function in WM because of its high expression? So then what we did, we used a database, a big database of many different tumors where there are RNA samples data available. So we are not going to go into the details of what kind of tumors, but what I would like you to focus on is that here, red represents tumors and gray represents healthy tissues. So you can see in this first set of tumors, the Wing2 expression is actually reduced in the tumor compared to the healthy tissue. But if we looked at this second set, then the question is different. It's actually the opposite. So the red goes up. So Wnt2 expression is actually really high in the tumor compared to the healthy tissue. So then this prompted the next question. So what are the mechanisms that actually are at play in WM that induce the increased expression of Wnt2? And we particularly focused on gene isoforms. So let's go back to cell biology for a moment to understand this concept of gene isoforms. So the DNA is the big storage of all the informations that allow every cell in our body to work. But then when a cell needs to function, it needs to take some of the information from the RNA, make some copy of specific genes at the RNA level, and they use that information to create proteins. And proteins are basically the functional unit in a cell. Then if we look specifically into the RNA at the beginning comes sort of in a raw format and needs to be edited. And this editing process is called splicing. What the splicing does is that it takes all the important pieces that make a gene, that are called exons, and here are represented in color rectangles, and they reassemble them in different ways. This basically allows for different types, different forms of the same gene to actually be produced. And those are what are called gene isoforms. But we can also think about them as sort of different gene flavors. And then those are used to produce protein. And so what it means is that the different proteins that are produced are different flavored, right? Flavored differently, meaning that their biological function in the cells may be slightly different. So let's translate this to Wnt2. So we looked first at what sort of gene isoforms was actually known for Wnt2 in the literature, and we observed that what was known is that this gene is really big, and there is a functional unit that is called kinase domain that's present, and then usually these forms, when expressed, have anti-tumor function. And they do so by inhibiting one very important signal in the WM cells that is called ERK1 and 2. Then we moved ahead and we looked at our own data, right, in our own RNA-seq in WM patients, and we discovered something new. So we discovered some gene isoforms that nobody has described before. And those were all, so we actually grouped them into two different families that here are represented in magenta and in yellow. And they, as you can see here, are share a particular event. So both of them skip, right, don't have two exons, and then one family still keeps that kinase domain and the other one does not. So we then were able to model in WM cells the most expressed of the gene forms within the yellow family. And what we observed is that it was able to actually drive a pro-inflammatory cascade. And this pro-inflammatory cascade activated the NF-kappa B and the ERK1 and 2. They are pro-survival signal that actually works in favor of the tumor. And so we discovered a novel pro-tumor, also called oncogenic function for Wnt2, that nobody has discovered before. So then we went back to some of the tumors that I've shown you before. We selected one, and we observed the same trend that we saw in WM. So all these novel magenta and yellow isoforms were actually highly expressed in the tumor, but not present in the healthy tissue. And finally, we wanted to actually see whether we could pharmacologically target Wnt2, but then we ran into the limitation that as of now, there is no inhibitor that is commercially available that specifically targets and selectively targets Wnt2. So we went with what was available, which is another drug that doesn't target only Wnt2, but targets also 3 other members of the same family. And then we treated WM cells, and with increasing doses of this novel drug, we were actually able to induce killing of the WM cells in what is called a dose-dependent fashion. So the higher the dose, the more cell killing we were able to induce. So in conclusion, our findings revealed that that Wnt2 actually does not only work as an anti-tumor, but also a pro-tumor gene, and this is really context dependent. And in WM, it does have a pro-tumor function. Wing2 is highly expressed in nearly all mediated mutated WM in early stages of the disease, which indicated that it may actually serve as one of these second hits that we talked at the very beginning. The relevance of our findings extend beyond WM to other cancers, and finally, further studies are needed, and that's always the case, to actually develop novel drugs that specifically target Wing2. With that, I would like to conclude my talk by thanking my mentor, Steven Trion, and my colleagues, Zachary and Shalu, that have been incredibly instrumental to this work. All our amazing clinical and laboratory team that always work greatly in tandem. All our collaborators, and obviously the IWMF, without whom this study could not exist. All of you, all the WM families and patients, obviously, and their caregivers. And finally, I would give a shout out to Ohio by remembering the two inventors of Superman, to actually remind every one of us that when we work together in tandem, we can be superheroes. Thank you.
It's so now I'd like to ask Hao-Sun to come up, and he actually just recently joined our lab a few years ago and has just taken off, and will present his work on single cell RNA sequencing and ibrutinib treatment in Waldenstrom's. It's very exciting stuff. And we'll take questions for the whole panel at the end.
Good afternoon, everyone. My name is Hao-Sun. I'm from Bean Center for Waldenstrom Macroglobulinemia at the Dana-Farber Cancer Institute. It's a great honor for me to present a summary of my ongoing project supported by the Robert Kell Career Development Award. The title of the project is Understanding the Dynamic Co-Evolution of Tumor Subclones and Immune Microenvironments of WM under ibrutinib monotherapy from a prospective clinical trial. Ibrutinib is the first FDA-approved BTK inhibitor for WM. It has helped many WM patients achieve meaningful clinical benefits. However, patient responses can be quite different. Some patients achieve better durable remission for a long time, but there are many patients relapsed relatively early. In many relapsed patients, we could detect alterations in BTK signaling pathway or downstream pathway, especially BTK mutations. But importantly, not all relapsed patients could develop the BTK mutation. So the central question in this project is, why do WM patients respond differently to ibrutinib in addition to BTK mutations? To address this question, we conducted a prospective clinical trial of ibrutinib monotherapy in WM patients. We followed the patients with a median follow-up of 5 years, and we collected the bone marrow samples before and during ibrutinib treatment, including the before treatment and post-treatment at cycle at 6 months, 1 year, and at 2 years, 3 years, and 4 years. For each sample, we conducted the single-cell RNA sequencing and the B-cell sequencing and T-cell sequencing, which allowed us to study both tumor cells and immune cells from the same WM patient at the single-cell level. First, when we look at the tumor cells, we could observe that WM tumor cells is not made up just one uniform single population. Different tumor subtypes could coexist in the same patient. Here, I'm taking one WM patient as an example. The 5 plot subpanels show the cell subtype, BCR clonotype, tumor subclone, and tumor subcluster, and the developmental pseudotype of the B cell or the plasma cell compartment. The first plot on the left is the cell subtype. The cell subtypes included the pre-B cell, memory B cell, and the malignant B cell and malignant plasma cells. These cells followed a linear developmental trajectory. When we look at the BCR clonotype, we found that the tumor cells share the same BCR clonotype, which means they originated from the same mutated tumor cell. Interestingly, in this patient, we found his tumor had two different BCR clonotypes, which means he may experience the two different independent WM tumorigenic events. Within the tumor 2, we further identified the 3 tumor subclones, the clone 1, clone 2, clone 3. They have totally different copy number variation, the CMA pattern. In clone 1, clone 1 had no copy number variation, and clone 2 had a deletion of 6q and amplification of 6p. Clone 3 further got a deletion of 20 based on the clone 2. These 3 tumor subclones followed a linear developmental trajectory from clone 1 to clone 3. When we track these 3 tumor subclones along with the BTK inhibitor ibrutinib treatment, we found that the more evolved the tumor subclone with more CMA events appear to be more resistant to ibrutinib. After ibrutinib treatment, these 3 tumor subclones behave differently. The early clone, clone 1, shrink, while the late tumor clone, clone 3, expanded. Importantly, we could observe this shift of abandoned tumor clone as early as cycle 6, that is 6 months after treatment. This is nearly 1 year prior to the clinical progression, disease progression was detected. Overall, we could identify 3 major patterns of how tumor subclone change over time. Here I'm using the 3 colors to indicate, to represent the 3 tumor subclones. The blue cells represent the early tumor subclone, while the purple cell represents the later, more evolved tumor subclone. The first pattern is devolution. This means the tumor subclone, the tumor became simpler over the BTK, over the ibrutinib treatment, and the early tumor subclone, the blue one, became dominant. The second pattern is no evolution, meaning that the tumor composition stayed relatively stable post ibrutinib treatment. The third pattern, evolution, means the tumor became, after post ibrutinib treatment, the later tumor clone, the purple one, became dominant, and the overall tumor composition became genomically more complex. The first 2 patterns, devolution and no evolution, were associated with the decrease in the tumor burden. However, the evolution pattern was associated with first an initial decrease followed by an increase in tumor burden. Further, we found these 3 tumor subclone evolution patterns were closely linked to the treatment response. The patient with devolution pattern appeared to be develop more favorable responses to ibrutinib, while the patient with evolution pattern tend to have poorer responses to ibrutinib. More importantly, in 3 out of 4 progressors, we could detect the evolution pattern prior to the clinical disease progression was detected. This suggests that using the single cell or genomic approaches could provide us an earlier way to detect responses to ibrutinib. So this asks us a question. If we can detect some certain signal as the baseline from the expanding tumor subclone like the purple cells to predict the responses to ibrutinib before the treatment. To explore this, we compare the expanding tumor clone versus the shrinking tumor clone at baseline and identified a baseline tumor subclonal tumor signal. We found this, we call this as WIP score, the Waldenstrom's ibrutinib predictor. We found the patients with a higher WIP score tend to have a greater risk of disease or progression. So this predictive tool may help us to identify high-risk patients at baseline. It's more earlier. In addition to tumor cells, we also looked closely at the immune cells. We first look at the T cells. This is an important part of the immune system. We labeled the different subpopulation of T cells. And we also use the TCR clonotype information to identify the some certain T cell population expanded from the same T cell clone. We found that in some patients, some certain CD8 positive effector memory T cells expanded post ibrutinib treatment. Further, we found these CD8 positive effector memory T cells also showed some difference between across the three tumor subclonal evolution patterns. We found the patients with evolution pattern have lower T cell cytotoxicity and elevated the PD-CD1 expression. The PD-CD1 encodes the PD-1, which is immune suppressive marker. This suggests that the immune system characteristic, especially the T cell functional states, could also influence the responses to ibrutinib. In conclusion, through the longitudinal single-cell analysis from both the tumor cells and their surrounding immune microenvironment cells, we found the variable responses to ibrutinib is shaped by both tumor cell subclonal evolution and the immune characteristics. We think understanding both aspects could help us identify the high-risk patients and ibrutinib earlier. In the future, this ongoing project may also, we think this could help us identify more additional immune therapeutic targets, including the targets related to T cells, the expanded T cells, and monocytes. Last, I would like to express my gratitude to my mentor, Dr. Chiang, and to all my colleagues at Bean Center. Especially, I would like to thank IWMF for supporting this research and making our research possible. I would like to also thank all the WM patients and their families for their participation and support. Thank you.
Thank you so much. So we are sort of right at the end of the session time. So can we do a couple of questions? What do you think, Paul? Yeah. There we go. So some quick questions for the panel.
Listen, I've got flats on. I can run. Okay.
Hello, I have a question about the Wink-2, if I remember correctly. Is that could that be a marker for either diagnosis and/or clinical treatment?
No. Can you hear me? Yes. Okay, great. Thank you. So yeah, that's a great question. So actually, because Wink-2 is specifically upregulated in PCL, and it's downregulated in BCL. There is actually some work that Dr. Hunter is right now moving forward with, that it's related to combining some of these potential markers, including WINK2, to actually do initially some better classification of the WM patients. So combining, since as you've seen, WINK2 is not uniformly expressed either highly or lower, right? I mean, if you take the PCL subtype, then definitely most of the people are in the very high end, but there are also few people that may be lower. So this raises the question and the possibility that actually we need a combination sort of therapy, and this is actually what Dr. Hunter is moving along. And I personally think is extremely promising because it would be a way to screen patient disease and better characterize it in a very accessible way, using a technique that's actually widely available throughout the world in laboratories throughout the world. So I think that's the best we can strive for. For disease response, I think it's still a little bit early for that. A colleague of ours a few years back published a paper about WINK2 being involved somehow in the response to ibrutinib, but we didn't know very well the subtype at that time. So now with what we know, looking back at those results, the signature that we got, it's probably related to a response that actually more subtype dependent rather than being related to WINK2 by itself. So we need to do more work.